So yeah, TMC, how are you tonight? We've got a full plate here tonight.
Just as you asked me that, I scrolled away from the window. I'm doing good, Indy.
Good.
We'll get synced up here soon enough.
So the overview of with the survey that's been going on for the last few weeks is topics such as the very smart smartphone, and we delved into that a little bit. Then 3 weeks ago, we delved into the mind, another domain of warfare. Could it be? Then we did part 2 of that, the mind as a battle space, future battle space. Then we started looking last week into nanotech. Exactly how small is this stuff? And it's really, really small. And that brings us to, as we get smaller and smaller, we start getting into the microscopic world of nerves, brain cells, and even DNA. So if you've been finding this of any remote interest whatsoever so far, I think it's going to continue for you tonight, because these things kind of, the nanotech and biotech are quietly merging together, and there's a whole lot of combining of the two. And believe it or not, this is becoming lower in cost the more that they perfect the science, the materials science, and the mass production of some devices, to the point where even devices at the level of consumer electronics are becoming smart enough to be able to complement and connect with these technologies, and perhaps ultimately to modulate them, but at the very least to act as an interface to other intelligent equipment. So you've got, in other words, you've got bridges. The bridge, we've got, we're going to be talking about some of the chemical and electrochemical effects and some of the compounds and molecules that have been designed for this purpose in the body. And then we connect through that by way of, sometimes it could be consumer devices, and we talked a little bit about that in past episodes. And that bridge connects to the outer world of intelligent electronics and equipment. So the possibilities are really huge. And I say this every week, and the possibilities really are huge. So what I'm going to do, if we would like to, is I'm going to dive right into a little video, and we're featuring videos from a number of other esteemed and well-accomplished scientists. Some of them are from the European Molecular Biology Laboratory. One of them is Ed Boyden. We have another one with a man named Steve Ramirez, and I'm not sure how to pronounce her name. I think it's Xu Liu. And so we're gonna kind of go back and forth between that and looking at some of the substantiation of what these material devices are, and how the science has progressed. So. TMC, any thoughts before I play that first one? I think you know which one it is. It's a short one, by the way.
No, I've got all kinds of literature, so I'll add as necessary.
So this is just a few minutes. It's going to kind of give you a little bit of an overview. Since we have acknowledged the producers and creators of this, I'm just going to let this run and see what you think of this. It's this introduction. Oh, by the way, what you're seeing here, we're talking about optogenetics. We're talking about optical electronic or optical electrostatic or optical magnetic or even optical thermal devices. Very, very, very small. What you're seeing on your screen right now is a picture of a neuron, and those little green dot things on there are the small devices which have attached to the outer surface of that neuron. And so here we go.
The brain is made of maybe thousands of different kinds of cells called neurons that are built into very dense intermesh networks that communicate. Each of these neurons computes using electricity. This cycle implements behavior and thought and emotion, all these different kinds of things. We also think that deficits in these electrical computations underlie many brain disorders that affect over 1 billion people around the world. In optogenetics, what we're doing is we're putting molecules that convert light into electricity into neurons, the cells of the brain. Then when we shine light on those neurons, light gets converted to electricity and allows us to turn on or off those cells. The goal.
So he's speaking very quickly here, but what they're essentially designing is a transducer. Changes one form of energy into another one, and it happens right at the point of interest, which is the surface of a cell, in this case, a brain neuron. And he goes on.
Here is to find a way to control the electrical activity in some cells and not others in the network. To do that, we had to turn to the natural world. It turns out that throughout all the kingdoms of life, in plants and funguses and bacteria and so on, you can find photosynthetic or photosensory molecules that convert light into electricity. So we borrowed these molecules from nature, and then using tricks from the field of gene therapy, we can put them into neurons. Now, these molecules...
So it's really very skillful in the process of studying what occurs in nature already, a little bit of reverse engineering and finding out exactly what types of molecules there are in these. Let me back it up just a little bit.
He said,
in this is a living creature, self-propelled, as you could see. And it sure looks like it's got a whole lot of some sort of photoactive material in it. And I'm not sure that they didn't actually add some to this, or that this is just an animation to try to prove the point. I am not so sure that there is a living cell that is that chock full of that many pellets of stuff, to be honest with you. I've looked at a lot of cells in a previous lifetime, and they don't usually look like that. But he's proving the point.
We borrowed these molecules from nature, and then using tricks from the field of gene therapy, we can put them into neurons. Now, these molecules can convert light to electricity, and they do it just in the neurons that we want to control, and not all their neighbors. So we can deliver these molecules to some cells and not others, and then when we shine light on the network, we can turn on or off that subset of the cells. If we can turn on or off a set of cells that's embedded within this dense matrix, we can figure out how do they contribute to a behavior. For example, if we can turn on a set of cells, we can figure out what kinds of behaviors can it initiate. If we can turn off a set of cells, then we can delete it momentarily and figure out what it's necessary for. So by being able to dial in information into cells in the brain and to delete them, we can try to figure out how they contribute to networks and the behaviors and diseases that arise from brain computations. We can...
Yep. So there are a whole lot of brain neurons in our heads, and they're connected by these little fibers. We've reviewed this in previous shows. This particular one, by the way, is from MIT, and this is Ed Boyden. You take a look at that right down there, from Massachusetts Institute of Technology, and he is a professor of biological engineering and brain and cognitive sciences. And this isn't new.
This was almost 10 years ago.
Yeah, it's gone a long way further, and he's got a lot more experience now. So his goal, and you know, they zeroed in on it, is to get these I'm just going to call them temporarily, just call them transducers, attached to specific cells, and then turn certain ones on. And you know, you get one of them. Let's say this one that my mouse pointer is hanging around right now. If you get that one so that it's inhibited, then all of the ones that it's connected to are going to be affected by that somehow. Or if you get 3 or 4 of them all inhibited or all stimulated, then they can study the effects on the periphery of this aggregate of very, very proximal neurons that you're seeing in this picture. So anyway, that's so it's important for them to be able to get them where they want them, and then for them to be able to very precisely shine the light that stimulates the effect on these designed molecules to do what they do, which in this case is either to stimulate or inhibit the cell. So that's what you're seeing, and keeping in mind that just as so many other things have become very miniaturized, yet they're very effective and very functional, that's exactly what's happening in this science. Smaller and smaller, more and more efficient, and more precise. Let's let him go on a little further here.
Hunt down the exact set of cells that are contributing to a specific disease state, or which, when activated or shut down, will remedy that disease state. That's really important because right now a lot of drugs are developed that target molecules, but molecules are found throughout the brain, and in fact many.
And I could just, and we could both, you know, just give a little bit of a clarification of that. When I guess I would say the classical pharmaceutical industry comes along and tries to treat something, they do their best to design a chemical or a drug to have a chemical affinity for a certain type of cell, but it's not it's not accurate at all. All right. It's a scattershot approach. If they're trying to, let's say, relax or lower the activity or the hyperactivity in a brain, that's just about the best that they're going to do with that drug that they'll design. Is it'll have an inhibitory effect on large areas of the brain, and that's different from what these guys are doing. They're trying to specifically target certain areas, and you know what? In terms of the kind of chemical toxicity and the overload to the rest of the body that some pharmaceutical drugs have, this is a far better way to do it. In some senses, it's more elegant, and they're working on it. So he's about halfway through this little explanation now.
Cells in the brain might be very molecularly similar to one another. If we can target circuits in the brain, though, we might be able to develop much more specific drugs. Imagine if we could hunt down the exact set of cells in the brain that, when activated, remedy a brain disorder, and then we can go in and look at the exact molecules in those cells. Maybe we can find drug targets that are much more specific than existing ones. You might
not only finding drug targets, but as you can see, it's working hand in hand. They will take what was the dark ages of the chemical pharmaceutical industry in attempting to use a drug for a kind of a curative effect in a brain, and they will pre-load certain brain cells so that they'll have a much more receptivity to that chemical if they put it in. And in fact, in some iterations of this technology, they're even using nanoparticles to deliver the actual chemical compound right to the site.
Yeah, you're talking about chemogenetics. It's a variation basically, but yeah.
Yep. I'm going to turn my volume up a little bit here so I can hear you. Okay. Yes, exactly. Yep. Chemotherapy, very, very oblique way of treating a disease. You're targeting just certain cancer cells with chemotherapy, but what happens? It puts a huge strain on the liver because it goes everywhere. They inject it into a blood vessel and it goes through the liver, the kidneys, the heart, the muscles, everywhere. It goes almost every place but exactly where you want it to go. And it is, it's kind of a double-edged sword because they need to use enough strength of classical applications of chemotherapy to be able to wipe out the cancer cells, but they can't use so much that they wipe out the parts of your body that you need to stay in good shape. So it's very, very hard with that. And so this is actually a welcome advancement in some senses. Now, you can also connect dots in other ways, and you could say, well, there could be some harmful effects to this too. Yes, there could. So, it's not about the technology. It's about who's using it and what they're using it for. And we let him go on and let him finish this thing up.
I also imagine that we could use optogenetics to directly control brain circuits in patients with brain disorders. Electricity is used to stimulate the brain in deep brain stimulation. If instead we could actually aim light at certain cells and turn them on or off, we might be much more specific. Rather than using electricity to turn on and off the cells of the brain and have many kinds of cells activated, the ones we want to affect as well as all their neighbors, if we could make just one disease-associated subset associated with light, and we can turn them on or off, we might be able to treat them with much more specificity. So far,
I'm trying to think of a good analogy for that in the macroscopic world. Something that's precise and elegant. Right? It's like a fire hose versus a pinpoint spray. Something like that, targeted, aimed precisely, and perhaps multiple pinpoint attack on certain misbehaving cells.
Optogenetics has had a lot of impact in the scientific world, but it hasn't been used in any human patients yet. There are a couple reasons why. One is that it requires a gene therapy to deliver the gene that encodes for these light-activated molecules into the body. Currently in the U.S., there are no FDA-approved.
So he said that very quickly, but let's just slow down on that. He used a couple of terms, gene therapy, and that there are, and he of course, this was in 2013, so maybe that has changed now. they do struggle to get FDA.
There's no
approval.
maybe about it. It has. You have emergency authorizations for all kinds of genetic delivery systems right now. So everything he just said no longer applies because we're in the proverbial pandemic. So,
but there's a difference between an emergency authorization and an actual approval.
That's right. That's right. But they're still able to deliver it's gene therapy, basically.
Exactly.
In many different ways, many different types of gene therapy. But what he just said back in 2013 was that
it was almost
it's not
predicting
possible
the future
in America because the FDA has not approved genetic therapy. And they have now currently approved specific genetic therapies, but they have also approved under emergency authorization, carte blanche genetic therapy for protection against, the most deadly pandemic ever, right? 99.99 or 99.97, percent.
I'm glad that you said it that way. I am hopeful that our video will stay up.
Screw them.
But yeah, no, I mean, we're not making any real commentary about it, at least not right now.
No, we're simply making the correlation that genetic therapy, or we're trying to understand and define what genetic therapy is and clarify that from 2013 up until present day, genetic therapy is in fact a useful, applicable, in circulation technology up to and including use under emergency authorization in current day for treatment for a pandemic. That's all, nothing more.
That's all. That's true.
Gene therapies. In Europe, there's just one. Another issue is that these molecules come from organisms like algae and bacteria. And so if we are putting these molecules into the body, would they be detected as foreign agents and attacked by the immune system, for example?
That's another thing. I don't want to really comment on that, but I would encourage people to just consider that when a foreign... When any sort of a molecule or a cellular component or a foreign body is introduced into the human body more often than not recognizes it as an invader, and that causes a reaction. And sometimes the reactions are very, very strong, which can have complications of their own.
He's delivered other talks about optogenetics between 2013 and present day, where he said that the majority of the pieces of genetic code that they're extracting out of the animal and plant kingdom actually are uptaken and adopted by human cells very, very well. There really is minimal to no rejection. They do exactly what they are supposed to do with minimal rejection.
The complexity and the commonality of life, both at the same time, right?
Yeah.
All right, his last 30 seconds here.
What we need is a paradigm shift in how we think about treating brain disorders. And one of our major stances is that we need new technologies if we really want to either understand the principles of treating brain disorders, hunting down the exact cells in the brain that can help us treat brain disorders, or to develop new modalities, new forms of energy, new strategies for treating brain disorders by correcting the computations within the brain.
Exactly.
He said correcting the computations in the brain.
Yep. So changing those computations.
Yes, for diseases, epilepsy, Alzheimer's, great. But what about correcting computations in the brain for things like behavior?
Well, who's the judge?
That is the ultimate question now, isn't it? Who is the judge?
Right. Exactly. So, again, we have to, we have to look at this very cautiously. Yep. I can certainly see many instances in which the gate is open to the use of this technology for the purpose of either inhibition or stimulation, and then it can, like anything, can go wrong. It does happen. Usually we hope that it goes wrong a lot less than it goes right, but a misapplication of the technology could cause the wrong effect on a certain person and or even a group of people. So there are a number of areas where one should be cautious. He says that for the most part, these molecules are uptaken into the human DNA backbone or other parts of the cell with little or no side effect. He says that, but I question how long we've really studied it and how hard we've studied it. And it's nothing against Ed Boyden, but I know that whenever I'm working on something, I get very enthusiastic about it, and I want to just keep pushing it forward. And sometimes it might be wise for me to slow down, because that's usually when things sort of unexpected things happen. So I don't know. At the very least, it is modification of humans at the cellular and at the genetic level, at the very least. Now, is it good? Okay, could be. Yep. Modification, if someone has, what is that? That corneal lens replacement surgery that people have sometimes, it is. Yep, that and the other one for cataracts as well, cataract surgery. So, these are modifications to the human body. And so we keep skirting around this whole issue. And our job is not to try to make these decisions for everybody who's out there, trying to assess all of it. It is just to bring the knowledge of what there is, what they can do, what they're working at doing, and then we show you exactly what they've built that does do it. And I have another little video which is kind of like a very one step at a time type of explanation of optogenetics, and I think I might play it. Yeah, I got it right here. Okay, well, actually, this actually takes it a little bit further. This isn't quite so basic, but I think we can safely move forward a little bit too. So check this one out. All right, what you're going to see is the discovery that there were cells from a frog that had an interaction with light and the ability to exploit that for purposes of making the experiment succeed. So here we go. This one's a little bit longer. I'll probably stop this a couple times. We may dive off into looking at some of the actual patents for some of this stuff too. That'll be kind of up to us as we see how we go.
Is rhodopsin and an ion channel combined, so it was given the name channel rhodopsin.
Now, he already went a little bit fast, but rhodopsin is a pigment, and combining that with the other effect, we develop a phenomenon with the rhodopsin that it causes it to move I'll play it again because it was very quick. In fact, I'll go back to the frog. Here's the frog right here. All right, so here we go.
Succeeded in transferring the gene for algae rhodopsin to frog's eggs. These now produce the corresponding protein. It was a surprise to find that it is a light receptor, that is, rhodopsin and an ion channel combined. So it was given the name channel rhodopsin.
Okay, so what you have right here is an ion channel. That's what this is. This is, if you will, this is the surface, the membrane of a cell. All right, the yellow thing, membrane of a cell. This is an ion channel. And in this particular embodiment of what either the discovery or the genetic invention, because they combined a couple things together, they have made something which has a selective permeability based on whether light is shined on it. Am I stating that correctly, TMC?
Yeah, I look at it like a gate.
It's a gate.
Yeah, so you've got the yellow stuff is the cell membrane. Those are lipids.
Yep.
And you have half of them. You see that the tails of the lipids, they're facing each other. So there's a layer that like water and there's a layer that don't like water. And biologically, these two things match up together, and that's what creates the membrane to keep the stuff on the inside of the cell separate from the stuff on the outside of the cell. In order to get things back and forth, and in this case, in order for communication to occur, you need channels or gates in between the inside and the outside of the cell.
Which normally occur. Those exist normally, but they took it one very, very ingenious step further with this little device.
Mm-hmm. And so what they're talking about here is a light gated ion channel, so that when you shine light on this particular protein, it opens up and it will move positive and negative charges back and forth across the membrane, and that's how action potentials occur, which is how a neuron signals. When a neuron fires, it's a couple of examples of that would be,
I don't know how many chemists there are out there, but Fe stands for iron, and Fe by itself would be plus. So you could have Fe plus sitting around out here, and inside here you could have OH minus. Now, if it's H2O, then it doesn't have a real net charge. But if it's just OH and it's missing one of the hydrogens, it has kind of a negative net charge. So there's your channel, and that is the gate for those two things to meet or not meet.
I've got to clarify, just for the sake of posterity, the types of ions that we're talking about here are sodium, chloride, potassium, calcium, and these things are positively or negatively charged. There's quite a few, actually, and each one of them has their own specific receptor sites within these gates.
They also have their own specific voltages too. Yeah. So in the positive species, we'd have sodium, potassium, and calcium. Those would be three different ones. They all have slightly different voltages and different atomic sizes as well. And frequently, you find those combined with chlorine. So you've got calcium chloride, potassium chloride, sodium chloride, which is table salt. But when they're together, they don't have a net charge, but you separate them, and you're going to have your Cl minuses down here, and you're going to have your Na, which is sodium, pluses floating around out here. And they'll want to join, but they can only join if this thing lets one of them through.
And that's how you get the negative charge, the membrane voltage, negative 70 millivolts approximately. That's how you get that charge, is the separation between the positive and negative ions.
Yep, and you can see it right in this picture, because you count up how many of the little green minus blobs there are, and you've got a net negative charge on this side of the cell membrane, and how many red blobs there are here, a net positive. So the cell is actually electrified in some sense. It's almost like a battery.
There's a voltage potential that exists
across that membrane. That's right.
Correct.
And even more, guess what fats are really good for? Insulation, electrical insulation, oils, fats. I don't get too far off in the weeds on technical stuff, but those transformers that they have up on poles, utility poles, they're usually filled with oil, because oil is a good insulator so that the transformer doesn't blow up, and they also help to conduct heat away too. But oil is a very good insulator of electrical charge. So oils are like fats, and these membranes are made of fats, which happen to be pretty good insulators. Anyway, just saying. See what else they talk about here. I like the pictures. I like all the little green dots.
The remarkable thing is that the channel reacts to blue light. It opens up and lets positively charged ions flow into the cell. Optogenetics scientists use this mechanism by smuggling the gene with the building instructions for the channel rhodopsin into the genetic material of the nerve cells, so that this channel can then work inside the membrane.
Wow, look how they do that. When they smuggle that in, they're most likely doing it with a piece of RNA code. It could be DNA, but it's easier to do it with RNA. And it is a payload on something else, whatever it is. He doesn't go into how they do it cleverly, but you can do it by using viruses as the couriers for it. Because viruses they happen to have an affinity for being outside of a cell and getting into it. So if you put something into the virus, and the virus is going to do what a virus is going to do, it starts outside of your cells and it goes into them. So it brings it in and then it releases it. And we've illustrated this a little bit a couple of weeks ago. Okay, what else is he going to show us? I love these diagrams.
If it is stimulated by light, positively charged ions flow into the cell. This changes the tension across the membrane, and the neighboring original channels also open up.
Oh, did you know that? It's a snowball effect. It's changing the charge down the line, down that cell membrane.
It's the axon.
Yeah.
Yeah, so it's creating that chain reaction on the axon, and if enough of those adjacent channels also open up and allow the membrane potential to change, then you actually get a complete
firing of the neuron.
That's right, a complete action potential.
Yeah.
Instead of a sub-threshold.
That's why it's drawn this way. That's why they specifically diagrammed it this way, as all these little minuses inside.
Yes.
But if you can picture just a wave of changing charge traveling right down and across and maybe even through that cell. Okay. That's good to know. So these are the mechanisms of how this stuff actually works. It's fascinating.
Charged ions flow into the cell. This changes the tension across the membrane, and the neighboring original channels also open up.
See the wave? There's a wave going right down the line.
An action potential arises. But the opposite effect also occurs.
It does have to reach a certain threshold to have that quote unquote action potential. Yeah. You could fill in the blank.
You've got to take it, and there's different amounts, but typically it's sitting at -70, and I think you have to take it to like a -30 millivolts before it'll complete the action potential cycle.
Yep.
Don't quote me on that, but it's I think it's just over half of the difference with the sitting at -70. I think it needs to be pushed to -30, which is a 40 millivolt change.
This is very much like wiring in electrical engineering. If you have a resistance, whatever the actual resistance of this whole structure is, you've got to have enough of a voltage change in order to overcome the resistance, basically. Let me go forward again here. We already saw that. Okay, he's up here.
Across the membrane and the neighboring original channels. This way, the cell is depolarized, and an action potential arises. But the opposite effect also occurs. Halorhodopsin, which comes from halobacteria, reacts to orange light.
This is also another really good diagram. It is a little bit diagrammatic. They don't actually look a lot like this, but in terms of the symbols of it, this part right here is the axon, and that's the transmitter. And these things that look like tree branches, they are called dendrites, and they're the ones that receive. And it's no accident you notice that these little receiving branches are very proximal to the transmitter of a previous neuron. And this is the way they connect. That is exactly how they do it. They look a little bit more squiggly than this. But that's the idea. Nerve cell, axon, dendrites, action potential, charge, another charge. And that action potential coming from the transmitting side off this axon being picked up by this dendrite, and it's sending that wave of change in the action potential down this dendrite into this neuron, which will then make that neuron fire, just exactly as the animation shows.
In this way, the cell is depolarized, and an action potential arises. But the opposite effect also occurs. Halorhodopsin, which comes from halobacteria, reacts to orange light and smuggles negative ions into the cell. This then deactivates the cell. Switching cells on and off using light, a fascinating prospect.
This is a method which is less invasive, as we say. In other words, it means I no longer need electrodes, which I normally always have to use in neurobiology. Of course, the implications are enormous. I can switch light on and off. I can make it work exactly as I like. I can even...
It looks like they have some sort of a prototype of that going right here, because I see many, many different little connectors coming in to this thing that is under an observation microscope. This is, it's not a real high-powered microscope. It's more like a dissecting microscope.
Stimulate individual parts of the cells separately without too much difficulty, and then can check on the signal.
So if you think of it, scaling this up would be scaling it up and knowing exactly where you've put these little active, well, I don't know, optically controllable, activatable gates. It's it seems like I'm curious how precise they are with the science of placing them now. That's where my mind is going next. Let's let it go a little longer here.
If we smuggle these ion channels into specific nerve cells, then one day we might be able to control diseased nerve and muscle cells in a targeted way. First of all, we breed them in bacterial cultures. To transport the genes, the scientists use viruses which they have reprogrammed, and which now...
Oh, okay. So it's I mean, that's just a commonplace thing. Just reprogram the virus. Pretty good. Yeah. Yep.
carry the information for channel rhodopsin or halorhodopsin. The advantage is that viruses are very efficient at transporting the information into the required cells.
Yep. Very good. Very good. Taking it all in. And
indeed, this nerve cell now produces channel rhodopsin, a light-controlled protein that originally came from green algae. In order to measure the electrical impulses, the optogenetics scientists carefully place a microelectrode in the cell membrane. They use light of varying intensity to try to activate the channel rhodopsin optimally within a single cell. It already works in the laboratory, but can it also be applied to living organisms or even in humans?
It already works in the laboratory 3 years ago. By the way, this one, I didn't call this one out, but this is from the Max Planck Society. This is March 2017, in case anybody was curious. And I'll probably want to put this link out there for you all, too.
If we want to transfer this method to human brain cells, for example, we would need to shine a lot of light. Because in order to reach the necessary nerve cells with the channel rhodopsin, the light would have to travel through the different cell layers. Only then would it be able to spark off an electrical impulse. But short-wave blue light contains a lot of energy and is therefore also dangerous.
If I work with very strong blue light, I may also cause damage to the cells. So it's all not as ideal as it might seem at first sight.
In order to solve this dilemma, the proteins must be made.
I think TMC, the way around that is to have light emitters, small ones, nano-sized light emitters in the tissues, right there in the tissues.
That's one way to do it. They're going to talk about another way where they actually dope or tweak the genetics a little bit and make it hypersensitive to a particular wavelength of light. So less light is needed to activate the channel.
Oh, you're talking about the rhodopsins. They are made hypersensitive so that they activate.
Yeah, so you can. Yeah, so it's like putting a turbocharger on a car kind of a thing. Where
they open the channel. They'll open the channel up way sooner than any other channel organelle would do so.
Well, with much less energy delivered, much less energy in that particular wavelength.
Got it. Okay.
Even more sensitive to light. We can achieve that by changing the selectivity of the rhodopsins. That means they should let a wider range of ions pass through the channel into the cell, or so the scientists hope. To do this, they change the genes, and hence the structure of the channel rhodopsin.
And that would be something that, in order to change the genes, I think you can extrapolate from this that when they're talking about making changes, these are kind of minor changes to genes, but they know how to do it, and they know how to deliver them.
This is highly sophisticated. So look at it this way: it's like they cut and paste genetic code from an algae, for example, into
a frog cell,
a mammalian cell, or an amphibian cell, right?
Whatever it is. Yep.
And so then they get their readings, and they understand how much energy, how they have to pulse it, all of that stuff, right? Now they're going to go in and they're going to genetically tweak the protein structure itself of the channel rhodopsin so that it's hypersensitive to light. So it's the only reason I interjected is basically to illustrate that this you're going on to a mastery, if you will, or another technical layer of genetic manipulation at this point, when you're starting to actually tweak the genetic code of the protein structure itself, rather than cutting and pasting it in a rudimentary way from one species to another.
Couldn't there be bad things that happen? I'm just saying, couldn't there be bad? Because this show is for entertainment purposes only.
We're going to find out in the next year. Did I just say that out loud?
Next month, even maybe. If you make a designed rhodopsin channel sensitive, hypersensitive, then won't it become harder for the cell itself to stay under control? Because there are naturally occurring other energies that come along too once in a while. So a natural channel rhodopsin has evolved over millions of years with a particular level of sensitivity for a reason. It's the right amount of sensitivity, but it's not so little sensitivity that it won't work. If you come along and make something that is more sensitive so that you can get the desired reactive effect with the light, in the laboratory from the outside, then that's not what nature did. So nature is not well adapted to that. I'm certain that it isn't, because it adapted itself over such a long time. In other words, hypersensitive ion channels.
Yeah.
Potentially.
But that ion channel would be impervious to heat, would be impervious to sound.
If they designed it that way, if they designed those sensitivities out of it.
It wouldn't, if those sensitivities, that particular protein structure is designed to respond to light. So if some other energy form, electricity or sound or heat, were to be introduced to that particular protein structure, it would do nothing.
It won't act. It's got to be the right frequency to ring its bell, so to speak.
It's got to be the right. It's got to be the right energy source.
Yep. I'm getting it. It's fascinating.
Causing a point mutation, the characteristics of the protein are suddenly different. The modified channel now allows more calcium ions to flow through the membrane, and the positive charge links it to the membrane. Now the cell reacts more sensitively to changes in tension. That means that the cell with a point mutation on the right reacts with an electrical response to much less light.
It's a huge advantage.
Okay, so he's taking it to the next level. He's making a super sensitive, is really actually making the whole nerve cell super sensitive.
Hypersensitive, yeah.
Yes. Mm-hmm. Mm.
And that's where if you were to take that idea, and if you were successful with that, now you can introduce something like an upconverted nanoparticle and have very weak photon emissions, relatively speaking. A piece of nanotech emitting a photon versus a laser being shined on a nerve cell. And you could see where that application would be useful.
Oh, absolutely.
Where you'd use nanotech, and you would phase the nanotech with energy, and it would upconvert and emit a blue photon, which they have that technology that exists. It's an upconverted nanoparticle. So if you were to make the channel rhodopsins hypersensitive, it may be enough of an energy emission in order to get a reaction to occur with something like nanotech inside the body.
Yep, and everybody remembers, I'm sure, from last week, the process of upconversion is you shine one color of light onto this molecule, and then it radiates a different wavelength of light back out. That's upconverting.
Yeah, I'll bring up a document.
I'm sorry, I did it to you.
I'll bring up a document real quick, and that way it'll be ready. We could talk about it real quick.
All right.
When we're ready.
By allowing me to penetrate more deeply into the brain. It's also interesting for numerous applications, for example, in muscle physiology.
Biomedical experiments have already shown heart cells that react to light. So maybe one day channel rhodopsin will be able to function as a sort of cardiac pacemaker. Other scientists have also carried out experiments in optogenetics with blind mice. Here they are swimming around aimlessly. But this mouse is now swimming towards the light because channel rhodopsin has been implanted into the cells of her retina, and she can now at least distinguish between light and dark. Activating cells using light-sensitive ion channels is one way of influencing the nervous system. But Professor Bamberg and his team are also investigating how to switch off targeted cells using light. That could be important for diseases like epilepsy, for example. Hello.
So I was thinking about the blind mouse, and that they incorporated channel rhodopsins in there, but if they could do it precisely enough to get just the right thickness of a coating of those on the inner surface of the retina, then eventually the brain would start to interpret those signals coming from that retina, and it would start to see. It would not just light versus dark, but it would start to develop an actual image out of it. They would have to be deposited very, very precisely in order to accomplish that, I think. Just thinking.
Rhodopsin is one very promising switcher off. The orange light paralyzes the nerve cells of the worm. It switches them off. The research work of Ernst Bamberg and his colleagues is being extended by teams of scientists throughout the world, who are working on heart cells, worms, insects, and mice.
As a scientist, I don't tend to...
Say again?
I didn't say anything.
Oh,
that
was the translator. Yeah, I'm hearing two voices, and I think one of them is you, but it's not. It's the translator. Sorry.
To get carried away by dreams. But what optogenetics can bring about is that in basic research, we can discover completely new facts about the circuitry of the brain.
Finally.
Oh yeah, definitely. It could very well be used as it was said earlier, for the purpose of mapping out. You know, get these things in there, and then provide a certain limited stimulus, and then watch the behavior. Watch what that cell does, and watch what other ones it affects. That's what Ed Boyden was talking about. Yep. Again, precise targeting. So that's the kind of thing that I think is what's going to make it even much more the more precisely. They've already designed the compounds. They've already modified the molecules. They've already created a piece of genetic code to make a cell make some of this stuff. So as long as you've targeted the cells that you want to be the ones making this stuff, then you've got your precise target right there. So how do you get those particular little bits of genetic code just to the brain cells that you want to get it to, so that they're the ones that have this modified sensitivity and electrical characteristics?
You'd find the right receptor compounds or receptor sites that you're looking for, and then engineer your virus to seek out those particular locations.
Well, yes.
The virus receptor would match up with a cell receptor, if you will, where you have a lock and key, and you would do it through the delivery vehicle, and then the genetic payload itself would be only delivered to the target locations.
I guess I agree with you, and I only challenge it insofar as I think it's within their range of power to design these so that they will have an affinity for either, pick any type of cell, an epithelial cell, a kidney cell, or a brain cell. But as for which specific ones that they get them incorporated into, that's where I think they're not quite there yet.
Well, you need to watch a little more Ed Boyden, buddy.
Okay.
Because Ed Boyden talks about using monomers to expand a piece of brain, for example, so that they can individually itemize all of the independent subcomponents, all of the independent proteins, because each one of them have unique markers and unique functionality. And if you can understand how to interact with those things, then you get exactly what you're doubting that they have, which is very specific site selectivity. Like deliver a genetic payload and have it stay dormant in a cytoplasm, or have it stay dormant in a ribosome, or have it stay dormant in the nucleus for some period of time until along comes some other type of a stimuli, whether it be a change in pH or a change in temperature or a change in a hormone level, or some new piece of genetic code that gets introduced into that cell that all of a sudden acts as a triggering mechanism,
right,
to then activate what was dormant. That's the level of sophistication we're at right now.
Okay. All right. I think that's kind of what we do. At least that's what I do when we discuss this stuff. I learn. He's almost finished. He's got just a few seconds left here, and I will back it up just 10 seconds too.
Light control.
The basic research, we can discover completely new facts about the circuitry of the brain.
Finally, light-controlled proteins are a good example of how basic research can open up completely new fields. Who would have thought?
So hang on. See that thing right there?
Finally, light-controlled proteins are a good example of how basic research can open.
Right there. That is a micropipette or a microtube that they're using to be very, very, very close to this cell. And this looks a lot like a nerve cell. So that's the scale. Cell. And they've made a little probe, almost like a little injection needle, that's that small. Very, 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 very small. Anyway.
Open up completely new fields. Who would have thought that the membrane proteins of tiny single-cell organisms could play such an important role?
There you go. Max Planck Institute of Biophysics in Frankfurt am Main. Thank you very much, Max Planck Institute. We're weaving all of this together into a coherent body of understanding of everything around us, and it's a lot. So that's why we do, we concentrate on one area at a time. There's just a lot out there to see how it all relates. Anyway, that was the end of that one. 2014, and there you go. So I took the share down. It's all yours at this point in time.
All right. I'm gonna we'll refresh these upconverted nanoparticles since we talked about them a minute ago, and
yeah,
this is a document we looked at the picture last episode.
Yep.
And
I think we should review this. And then I've got some more videos with very simple diagrams, but this is good to tie it into the last discussion.
Sure.
Thank you.
So this is Kurzweil.net. Ray Kurzweil, futurist, he was basically counsel for companies like Google and the president, former presidents and whatnot. So this document here talks about using near-infrared light to penetrate through biologic tissue to excite little baby nanoparticles that then emit blue light. That's really what this is. And what this is talking about is a less invasive way to stimulate the brain with light in order to treat neurologic disorders. And it says the new method extends the depth that optogenetics, a method for stimulating neurons with light, can reach. With optogenetics, blue-green light is used to turn on light-gated ion channels in neurons to stimulate neural activity. Blue-green light is heavily scattered by tissue that limits how deep the light can reach and currently requires insertion of invasive optical fibers. So researchers took a new approach to brain stimulation, and they used longer wavelength, 650 to 1350 nanometer near-infrared light, which can penetrate deeper into the brain via the skull of mice. The near-infrared light illuminated upconversion nanoparticles, which absorb the near-infrared light and glow blue-green in formerly inaccessible, deep, targeted neural areas. The blue-green light then triggered via chromophores light-responsive molecule ion channels in the neurons to turn on memory cells in the hippocampus and other areas of the brain. These included the medial septum, where nanoparticle-emitted light contributed to synchronizing neurons in a brain wave called the theta cycle. And so this is really how it's done. Nanoparticles effectively extend the reach of lasers, enabling remote delivery of light and potentially leading to non-invasive therapies. Research group leader at the Rankin Brain Institute in Japan said. His name was Thomas McHugh. In addition to activating neurons, upconverting nanoparticles can also be used for inhibition, turn off neurons rather than turn them on. In this study, upconverting nanoparticles were able to quell or quiet experimental seizures in mice by emitting yellow light rather than blue to silence hyper-excitable neurons. And so what you see here in this diagram is near-infrared light is shined onto this piece of nanotech. This nanotech gets excited and it emits blue photons of light that happen to be the correct wavelength for this channel rhodopsin, which has been inserted in a mammalian organism or in a mammalian cell known as optogenetics or an optogenetic therapy, gene therapy.
Now, I have a question. I have a question. May I just ask? When it's near-infrared light, it could be near-infrared coming from the visible side or near-infrared coming from the microwave side. Is this coming more from the very deep red side, the visible side of near-infrared?
It would be dependent upon how the nanoparticle was constructed or how the channel, what wavelength the channel rhodopsin responded to, because they have channel rhodopsins that respond to infrared light without the need for any sort of conversion.
Well, you know what I'm getting at. Could they design these in such a way that they were somewhat responsive to something even lower, like maybe in the very, very high microwave band? Would that be possible, or is that outside of the scope of what you can do?
It would probably not necessarily fall in the traditional optogenetics. Optogenetics, you're talking about light. As we understand light, it's technically like visible light, or even infrared is technically considered part of light. But if these things were responsive to microwave or millimeter wave, it would be
Well, it's all electromagnetic radiation. So you know where I'm going with it is, I mean, you know how they build things. They make one and then they modify and pretty soon they've got a whole panorama of different flavors of something. And
it really wouldn't surprise me if they've actually been able to create protein structures that are basically make ion channels that are responsive to that. But I haven't personally seen anything like that. If a cell was to be stimulated with millimeter wave or microwave energy, it's typically done through a heat conversion process or through the conversion process of turning electromagnetic waves into sound.
Right. That's what it usually is. And that's the heat conversion process. But let's say that something was able to directly receive the microwave frequency just as if it had been stimulated by the infrared or the near-infrared light. Now you've got a whole different and totally invisible form of energy that you can use. And we do know that microwaves will go right through flesh. they'll go right through flesh.
Oh yeah. Oh yeah.
So as we move up the spectrum, what happens? Okay, so let's just draw that spectrum, but just very quickly. Microwaves, low. Get a little higher, it's infrared. A little higher, it's red. Get a little higher, it's green, blue. Then you get up to the ultraviolet and the violet. And when you get up to the ultraviolet, it penetrates less into the tissue, but it tends to burn the surface of the tissue more right at the surface because it doesn't penetrate. Whereas the lower frequencies tend to go right through things. So that's why I was kind of drilling down on that whole idea of whether they even have the idea of trying to design them to be excitable and controllable with those frequencies. I was just a little diversion, that's all. I was just thinking out loud.
Yeah, good thoughts. Here's another article real quick. This was from a while ago, 2018, nanoparticles in mice brains light up and trigger memories. And this article talks about optogenetics using nanoparticles to convert near infrared light to visible light in order to activate deep brain neurons. Yeah. So they talk about the different types of implants that they've used as this technology has evolved. This is a very interesting article to read. But here's this Japan's Rankin Institute again. Very, 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 very common to see this institute put when you're looking at optogenetics. But what they're talking about here again is near infrared light being used to stimulate inside the brain, and the near infrared light can come from outside the skin. So no implants necessary.
Sure.
They say specifically because near infrared light can pass through tissue. And so they talk about this experiment, and what they really did is they put infrared light in the brain, and obviously it converted to a different color in the brain and emitted those photons, and it started to activate nearby neurons. So the researchers tested the strategy in awake mice in a variety of ways, including an experiment in which they artificially replicated scary memories, causing mice to freeze in fear. The opposite, activating happy memories to protect mice from stress, can be done as well. So we're talking about mood and emotional manipulation.
I want a world full of happy mice. Come on. I don't want mice under stress.
Yeah, well.
Come on, man.
A psycho-civilized society is still a slave society.
They actually demonstrated that. They were able to show that they could, and whatever manifestation that a mouse has as fear, it'll do something. It'll twitch, its eyes will bug up, maybe its whiskers will stand up.
They'll freeze in place, or they'll run off into a corner and stay there. You know what I mean? I'm sure that they've studied the behavioral characteristics of mice at nauseum in order to understand whatever testing that they do on mice, which is a very standard animal to be testing on.
What's the date on this one that you were just you're reading from right now?
This is published in 2018, February 8, 2018.
Wow.
Yeah, and so it's
if you can do it with a mouse, you can do it with a monkey.
Yeah. And they're already doing this kind of stuff in the eyes of blind humans. Optogenetics has been approved for human use to treat specific types of blindness.
Yep.
So they're already at the human stages with this technology in general, with this particular technology in general. They're already at the human stage.
So I'm reading down there. Thomas McHugh says, I was the doubting partner in this. A neuroscientist at Rankin who collaborated, he said, I was like, there's no way this is going to be efficient enough, but he was surprised at the results. Mm-hmm. The seemingly impossible is what made Anakiba and Deseroth, the inventors of the technique, reluctant to pursue the line of work beyond patenting the calculations. But as you can see, they have tangible results in laboratory, in vivo, with mice.
It's in the body. That means in live mice.
Yep.
The composition allows lanthide molecules to be excited and reactive to the wavelength of photons in the near-infrared spectrum. So lanthide-based or lanthide-doped nanoparticles, they emit visible high-energy photons as they return to their resting state.
What I found a great diagram of all. I'm looking for a diagram right now. I'll get it, and I know where it is, and I'm going to put it up in the share after you read a little bit more of that. Let me locate this thing.
Yeah, this one was done. Let me find another one. I think we looked at these of converting nanoparticles. You could buy the stuff on the open market.
Yeah, we did. We did look at this. Yep.
Yeah.
That was last week. Yep.
Mm-hmm. I mean, these are, and they're food grade. I mean, you can consume them. They're completely food grade. There was another one in here that I wanted to show. Let's look at this one real quick while Indy's looking around for something. Stealth cap technology for light emitting nanoparticles. And
I've found what I was looking for.
Good.
I just, in order to get it over to the computer where it will display, I will need to pull the share down.
Do it.
Or someone will have to. Maybe you could do it.
Yeah, I'll drop share. It's just another cool little image where they're showing up-converting nanoparticles that are doped and encapsulated, flowing around in your bloodstream. So,
I may be able to do it while you have the share still up. Let me see here. Yeah, it's going to look a little sloppy, but
I'll pull. It's yours, buddy.
Right.
Take it.
Yeah, you know what? It's really hard to do. Darn it, it's on the wrong computer. Well, that's all right. I can transfer it another way. It'll just take me a minute. Okay, keep going. I know what I'm going to do. All right. I'll get it.
Very well.
It's going to take me like 2 minutes to get it.
You're good. Well, I want a couple things out of here that are useful to, I think, share. It says nanoparticles are tiny structures, typically less than 100 nanometers in size, which is about 5,000 to 10,000 times smaller than the thickness of a human hair. These materials are receiving increasing attention for biomedical applications. So it says here, it's been known for some years the distribution of nanoparticles in the body is essentially determined by their size and surface properties. Up-converting nanomaterials are of great interest for biomedical imaging. When stimulated with infrared light, they can send out bright blue, green, or red signals. If we succeed in navigating such nanoprobes to disease tissues, it can be particularly useful for cancer diagnosis. So there are some good applications for this kind of technology. You ever heard of a photochemist before, Indy?
No, but it's going to be a field that starts growing.
Photochemist. That's an interesting. That'd be an interesting word to look up. The light up converters show poor solubility in water or tissue fluids. It must have a feature before any diagnostic or therapeutic can be used. So what they did is they improved this stuff. They put a new cover on this stuff. And what they're saying here is that the research shows that the new cover they put on these nanoparticles can almost completely prevent the body's own substances from binding to the particles. In other words, the nanoparticles now seem to wear an invisibility cloak. They believe this will help avoid their recognition and elimination by phagocytes in the immune system. So what does this mean? This means that if they put this stuff in the body and the cells die and go through their regular replication process and the dead cells get excreted out and processed like waste, these little nanoparticles will not be looked at by the immune system and will stay in place and won't be excreted, won't be removed from the body. It creates a more semi-permanent placement where you don't have to constantly reapply nanoparticles.
It could
over and over and over again.
Could there's another thing that could happen too. They could also, some of them could get excreted out and wind up out in the environment, out in the food or the water chain, and then get taken up again by somebody else. That could happen as well.
Yeah, there's a lot of things I think they could do.
A lot of things that could happen, really.
Yeah, it's we are in a sci-fi world nowadays.
Right,
are you ready, or do you need a little?
Yeah,
more?
I know. I got it now. I just want to open it up, and I due to the nature of the way the computers are connected, I actually had to transfer it with a flash drive, which was very clunky.
So this was back in 2014. February 9th, 2014, new opsin is one of, they're talking about different opsins, and this article is related to Optogenetics and the fact that, they're talking about using genetic code out of blue-green algae. No, I take that back. They're about opsins that respond to light in the blue-green range. And this is the piece I wanted to show. I got a little ahead of myself. The new opsin is one of about 60 light-sensitive proteins found in a screen of 120 species of algae.
Oh, wow. Naturally occurring.
Yeah. So what this means is that there's 60 light-sensitive proteins that are potential candidates to choose from and or to study to understand common denominators. If you were going to synthetically engineer a light-sensitive protein, you would use these as a study group to be able to understand,
Great opportunity for reverse engineering.
Mm-hmm.
All right, so I got this little thing. Let me put it up, and it's a little bit off topic, but I think people might find it a little bit interesting. Let me just get it.
I want to share one more thing.
Okay.
Here's what really stood out to me in this article. They're talking about a red light sensitive opsin, which researchers have named crimson. I don't know if any of you've heard crimson before, but just look that up, crimson, around 2018 or 2019. I'm not saying there's any kind of relationship there. I just find it incredibly ironic that this name came up in the field of optogenetics as it relates to gene therapy, Operation Warp Speed and all that. But the red light sensitive opsin named crimson can mediate, or basically means manipulate, neural activity in the response to light with 735 nanometer wavelengths. Guess where that wavelength resides, folks?
That is infrared. That's red.
Near infrared range, less than 100 nanometers away from the center channel, the center frequency for your iPhone Face ID.
Oh, really?
It's less than 100 nanometers away from that. And the patents that you can find that relate to things like Face ID and the laser systems that do 3D mapping for your phone talk about having a variable infrared spectrum, if you will, to be able to understand and pass through things like tissue and do analysis with wavelength shifted calculations. And so
they can swing from, let's say, 700 to 770, and they can adjust that slightly.
Yes, they can swing more like 650 to 1100.
Okay.
And then there's other patents that are kind of in the same family, not from Apple, where they talk about using the same diode to actually send two different frequency bands out of the same diode, where you're pushing up into 1600 nanometers and then going as far down as 600. So the frequency spectrum that's plausible or possible was something like a flat substrated built into your phone behind a nice, sleek, sexy glass. There's a wide frequency band there that's available.
That's kind of what we said at the beginning of the show. It's just fascinating that the convergence of some of this technology seems to be converging around not only very low cost, but very, very plentiful devices that people have around them. And, that once again, last week we were talking about exactly how many laser LEDs actually project out into the environment from something as simple as a smartphone that everybody's got. Yeah. So let me digress just a little bit, just to take a little bit of a break and look at the world of different types of materials. So we're talking about material science here. And I found this a little bit interesting, and you might as well. And it kind of looks like this. So let me just we're going to take our time with this. And they assembled this little graphic for the purpose of studying the relative strength versus flexibility and lightweightness of different materials. And at one end of the spectrum, they've got things at the bottom end of this that are like foam. So foam tends to be very light, but tends to be very weak. And then at the other end of it, they have things like, so as from the green end is very, very low density, very lightweight, very pliable, and not really very strong. Now we go up to the other end of this, and we have ceramics. But the area that I focused on that was really interesting to me was the area right in the middle, where they we've I don't know how many polymers that we've talked about and how many composites that have a combination of a metallic molecule incorporated into something that might be an elastomer or even like almost like a plastic. And that's in this area right here. So combining from both ends of the spectrum, that's possible in a laboratory. I mean, you can make a compound that fits anywhere on this whole spectrum of very hard and very brittle and very dense to being very, very lightweight to something that can be small and still lightweight enough. And that's right in here, right in this area where we find that the polymers, the elastomers, and lo and behold, the natural materials tend to show up a lot here too. So right in this area, and I would submit to you that this is the area of polymer, plastic, and biomolecule science where they're really concentrating a lot, is right in this area, because these are the ones that you can get into living tissues, and you can incorporate things into them or onto them, just like you said, so that they can be cloaked. You can have your little metal particle, but you can surround it. You can polymerize around it with something that the body finds to be friendly, and it will slip in. And that's not even getting into the idea of designing it so that it has a particular charge affinity or a particular affinity for a wavelength of light where it might change its characteristics. So that's like the whole spectrum, you know, from soft and foamy to hard as a rock to right in here. And this is the territory of biomolecules, right in that area. You find that fascinating? I was kind of fascinated by that diagram.
Oh, yeah.
Anyway, that's all I wanted to show you. Have another interesting, I have a video with a real catchy title. It's a very catchy title. I guess with that introduction, I need to just play it.
Yep.
Okay, here we go. So the title of this is A Mouse, a Laser Beam, a Manipulated Memory. Okay, mouse, laser beam, manipulated memory. So this is basically, these guys are just going to talk about what we just read about in the experimentation that they were talking about, where they induced either fear or happiness. So here we go. Let's get this thing up there. And this is this is a Dr. Steve Ramirez and a lady named Xu Liu. I hope that's how you say her name. Okay. And this is from a TED Talk. Here we go.
So there is a catch. There is a catch. These ideas probably remind you of Total Recall, Eternal Sunshine of the Spotless Mind, or of Inception, but the movie stars that we work with are the celebrities of the lab.
Test mice.
Mice. It's mice.
Work in the lab with mice, trying to understand how memory works. And today, we hope to convince you that now we're actually able to activate memory in the brain at the speed of light. To do this, there's only two simple steps to follow. First, you find and label a memory in the brain, and then you activate it with a switch. As simple as that.
Are you convinced? So, turns out finding a memory in the brain isn't all that easy.
Indeed. This is way more difficult than, let's say, finding a needle in a haystack. Because at least, the needle is still something you can physically put your fingers on, but memory is not. And also, there's way more cells in your brain than the number of straws in a typical haystack.
Right. So the thinking, I think that the state of the art with the technology right now is to be able to target a cluster. And I think it's something like a pinpoint of about 1 millimeter, maybe a millimeter by a millimeter. Not very big, but that can be huge relative to the number of nerve cells that can be, under the focus of that target. But at the same time, all those nerve cells they're kind of like all birds of a feather, if you will. If you ever took a look at, I think it's Gray's Anatomy. If you took a look at Gray's Anatomy and you looked at the areas of the brain that are dedicated to different centers, different specialized areas of function, they're actually kind of large. So when you can get down to a millimeter size, you might not be able to pinpoint an exact specific memory verbatim or a specific thought and either stimulate it or attenuate it, but you can get in the ballpark, and that's where they are with down to that level of granularity of targeting right now. They can definitely target a center of the brain that is responsible for a general group of behaviors, let's say. So he's gonna talk about how they're working on it.
So yeah, this task does seem to be daunting, but luckily we got help from the brain itself. It turned out that all we need to do is basically to let the brain form a memory, and then the brain will tell us which cells are involved in that particular memory.
So what was going on in my brain while
And what that means is they get these optically active molecules in there, and then they just watch a person think, and they see what type of interaction it is with the artificial molecules that they've gotten into the brain, and then thereby they start creating a map. That's what he's saying.
I was recalling the memory of an X. If you were to just completely ignore human ethics for a second and slice up my brain right now, you would see that there was an amazing number of brain regions that were active while recalling that memory. Now, one brain region that would be robustly active in particular is called the hippocampus, which for decades has been implicated in processing the kinds of memories that we hold near and dear.
I think I have to apologize. I think I might have said that Xu Liu was a lady. It's not. It's a guy. My apologies. It was completely unintentional. Hippocampus. They're talking about the hippocampus now. Okay.
Which also makes it an ideal target to go into and to try to find and maybe reactivate a memory.
When you zoom in into the hippocampus, of course, you will see lots of cells. But we are able to find which cells are involved in a particular memory. Because whenever a cell is active, like when it's forming a memory, it will also leave a footprint that will later allow us to know these cells are recently active.
So the same way that building lights at night let you know that somebody is probably working there at any given moment.
That's a great analogy. Great analogy. Let's say this whole thing was the hippocampus, and then within it, they're just looking for, and once again, at that very, very granular, very, very optically precise level, they're looking, or electrically level, they're looking for a pinpoint of activity when a specific thought or behavior happens, or a memory is remembered.
In a very real sense, there are biological sensors within a cell that are turned on only when that cell was just working. They're sort of biological windows that light up to let us know that that cell was just active.
So we clipped part of this sensor and attached that to a switch to control the cells. And we packed this switch into an engineered virus and inject that into the brain of the mice.
Okay. Engineered virus injected into the brain of the mouse, and they've basically built a switch, and they get it in there, and they don't really need to know where it's going. All they need to do is just get it in there, and then they watch what happens while the subject specimen goes about its process of moving, thinking, eating, looking, everything that that a subject specimen would do.
So whenever a memory is being formed, any active cells for that memory will also have this switch installed.
So here's what the hippocampus looks like.
You have any comments about this, TMC? I mean, doesn't your imagination just really go wild with this? Mine does.
I'm trying to visualize how the mechanics of deploying this kind of stuff out into the mass population would be possible.
You know what the beauty of it is?
They're literally talking about putting stuff in the brain or the body and then watching it to image it to understand things like memories. I mean, don't you think that's right. That's a control freak's wet dream, man?
That's basically it. It's like it got a lot easier just by knowing that they could relieve themselves from having to as long as they can observe precisely, that's all they really need to perfect. As far as getting the compounds in there or out there by way of, exactly as he said, delivery by way of a virus with an injection, okay? That's the easy part. That's the easy part, TMC. Now, once you've got all that out there, now you can just sit back and observe and map it out. I think that's really, really clever in a dark kind of way. This is an entertaining show, though. So, yeah, these guys they're fascinating.
After forming a fear memory, for example, the sea of blue that you see here are densely packed brain cells, but the green brain cells are the ones that are holding on to a specific fear memory. So you're looking at the crystallization of the
Okay, so this is exactly what we were just talking about. Associated groups. You don't have to get right down to one exact single neuron. They're birds of a feather. They work in neighborhoods. And this is right here. I'm not sure what the magnification is here, but this is a neighborhood where a certain stimulus was applied, and these things are all activating, at least as he said, I'll play it again. He said as fear. Okay, here we go.
Hippocampus looks like after forming a fear memory, for example. The sea of blue that you see here are densely packed brain cells, but the green brain cells are the ones that are holding on to a specific fear memory. So you're looking at the crystallization of the fleeting formation of fear. You're actually looking at the cross-section of a memory right now.
So did you know that you could see fear, literally, with a flashlight? I mean, did you know you could see it?
I do now. I'm looking at
the level. Another thing we could add at the level of magnification here, we're probably at around like 250x magnification, and the neurons are really, they're visible, but they're actually very small. It's nothing like the high resolution picture that we were showing of other neurons where they zeroed. If you see my mouse pointer, just that one dot right there, that's what we were showing you before, was single neurons. But this is like groups of them. You could almost count, like there's every one of these little fibers coming along here is connected to a neuron somewhere along the line. And some of them are on the surface so you can see them, but others of them are at a deeper layer. So I'm just going to move my pointer from here to here. And I'm going to say there's probably a couple hundred, given that it's one mouse movement wide and probably the same mouse movement deep into the tissue, like moving further in depth, right? So, 100 by 100, 1,000, couple thousand, 2, 3, 4, 5,000, maybe 10, 20,000 neurons all around here in this whole thing. And they know what they're doing. And they're all kind of reacting the same way. Okay. I want to listen to this. I hope everybody, you guys want to listen to this? All right.
Looking at the crystallization of the fleeting formation of fear, you're actually looking at the cross-section of a memory right now.
Now, for the switch we've been talking about, ideally, that switch has to act really fast. It shouldn't take minutes or hours to work. It should act at the speed of the brain in milliseconds.
So, what do you think, Xu? Could we use, let's say, pharmacological drugs to activate or inactivate brain cells?
Nah, drugs are pretty messy. They spread everywhere. And also it takes them forever to act on cells. So it will not allow us to control a memory in real time.
So TMC, how would they do it? If drugs are too slow and they're chemically very diffuse all over the place, how would you do it? If you were right up there right now with these guys, saying, well, I suggest we do this, what would you suggest that they use?
Well, the fastest is light.
It's fast, really fast.
Yeah, the fastest would be light. And then
Pulse light? Oh, yeah.
You've got sound being also very fast. And then when you're talking about the distances from like outside the head into the head, the speed of sound is plenty fast to deal with millisecond timescales, in my view. And then you've got the heat, the opportunity for using heat
where you can actually do spot heating using a different form of energy to get the heat there, but then you create heat at a very, very tiny location very quickly.
It's you basically you're using photoacoustics or electroacoustics, and that's basically what that is, right? Is you're applying light or you're applying electromagnetics to tissue, and it's rapidly heating up, and you pulse it on off very, 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 very fast, and it creates sound waves because you get expansion and contraction that will create ultrasound in the body. And really what it's doing is it's heating up tissue and letting it cool. So you can use ultrasound all by itself, or you can use light or electromagnetics to generate ultrasound. And if you modulate light or electromagnetics a little differently, then you actually get a heating effect, which, I mean, the FCC regulates for a heating effect.
You can create the ultrasonics in that tissue either with just a pair of microwave frequencies or a pair of light frequencies. You can do it with there's many methods. I wonder which one these guys like. Many methods. All right. Maybe he'll tell us. I don't know.
So, Steve, how about let's zap the brain with electricity?
So electricity is pretty fast, but we probably wouldn't be able to target it to just the specific cells that hold on to a memory, and we'd probably fry the brain.
Oh, that's true.
Okay. It's not going to be electricity. No. Okay. Next.
So looks like we need to find a better way to impact the brain at the speed of light.
At the speed of light. That means radio waves, infrared, or light itself. I don't know. I'm not a scientist.
So it just so happens that light travels at the speed of light. So maybe we could activate or inactivate memories by just using light.
That's pretty fast.
And because normally brain cells don't respond to pulses of light. So those that would respond to pulses of light are those that contain a light-sensitive switch. Now, to do that first, we need to trick brain cells to respond to laser beams.
Yep, you heard it right. We are trying to shoot lasers into the brain.
And the technique that lets us do that is optogenetics. Optogenetics gave us this light switch that we can use to turn brain cells on or off. And the name of that switch is channel rhodopsin, seen here as these green dots attached to this brain cell.
There, so we're connecting all the dots. We're tying all the pieces together from what we had going earlier in the show. Okay. All right.
You could think of channel rhodopsin as a sort of light-sensitive switch that can be artificially installed in brain cells so that...
Artificially installed in brains. Now, guys, you see the size of these things, right? They're these little itty-bitty green dots. If we were to do a cross-sectional diagram of something the size of, let's say, a 22-gauge hypodermic needle, then you would have probably 100, and I don't know, but I'm just going to guess, 100 to 1,000 neurons across inside the barrel of that hypodermic syringe, okay? This is the kind, and I'd like to research that to make sure I'm right, but it's like a lot. In other words, they could be standing next to each other, all parallel with each other in the barrel of that 22-gauge needle syringe. And now we look at these little green dots here. Those are wicked small. Just to give you an idea of the scales that we're talking about.
Now we can use that switch to activate or inactivate the brain cell simply by clicking it. And in this case, we click it on with pulses of light.
So we attach this light-sensitive switch of channel rhodopsin to the sensor we've been talking about and inject this into the brain. So whenever a memory is being formed, any active cell for that particular memory will also have this light. So
there they go. They're talking about injecting it right into the brain. And that's a good, effective way to do it, as long as it's done carefully so as not to harm the brain. If there were something that were designed that is able to travel through blood vessels, and if it's able to cross the blood-brain barrier, if it was designed chemically in such a way that it could cross the blood-brain barrier, then you wouldn't necessarily have to inject it directly into the head. You could still get it. Probably would go everywhere. Let's say that these very, very small little thingies here, these little green thingies, they might have gone all over the body, right? But the idea is light is going to be shown on very specific areas of the cells where these things got attached to. And these would be designed in such a way as to have an affinity for that type of cell. So they'd just roll right by muscle cells.
I gotta add something here.
They'd just roll right by epithelium cells. If you drank them, they'd just slide right down your throat. But
I gotta add something, Indy.
All right.
Yeah. So don't think that this isn't possible simply because you hear them talking about sticking a needle through the skull of a mouse brain and directly injecting genetic code into the brain, directly putting a syringe into the brain. Now, when you think about research funding and you think about efficiency, how much does a mouse cost versus developing technology to insert something into the vein or into the muscle of a mouse and have it travel up to the brain and deposit when a mouse lifespan is going to be over when the experiment is over? Why would you do that? You wouldn't. You just shove a needle in the damn head of the mouse and you inject the code directly.
You've just posed a lot of questions. First question that you asked is, how much does a mouse cost? I honestly don't know how much a mouse costs.
It probably costs a lot less than years and years of R&D to develop a system that can be delivered either through the vein or through the muscle to then be transported up to the brain, cross the blood-brain barrier, and then find the particular brain center of interest. That would be much more expensive than a mouse, one mouse.
Yeah, what they're doing is they're pushing to the side some of the other challenges for now, and they're saying, right, all right, let's get some mice, they're cheap, and we're just going to inject this mouse right in the head. All right.
Right. They're looking at it a piece at a time. They're not looking at the holistic. Right.
I get your point. Yes.
Okay. Just wanted to because some people I got stuck on that personally when I first started looking years ago. When I first started looking at optogenetics, like as a thing, I'm like, well, this whatever. I don't care about this because everything I was reading is we injected this into the brain. I'm like into the brain, thinking to myself, and I'm looking, I'm reading, and yeah, they're putting syringes through the skull into the brain of a mouse, or through the opening in the skull, which they then attach a fiber optic to stimulate the brain with light. And then you read on further.
Within the range of the science of biochemical engineering now to design particles that will have specific affinities. Targeted
gene therapy, targeted delivery of nanoparticles. These are all very well known, very well studied, very well developed technology. So you just basic, put those two together.
They're already doing it.
So you have a delivery
right now today. They've done it. I mean, I won't say it on, I'd like the show to be able to stay up and not get taken down, but I mean, they're doing it, 10,000 people a day right now. I'm not going to say what they're doing, but they're doing it.
You have a delivery system and you have a payload system. And then you have the science of being able to put those things together where they're stable and they function properly in the target of interest in vivo. Yes.
While it stays alive. That's right. That's right. And they can just park there. Now, you think these things really cause a whole lot of harm just being parked there? It's just, they're just attached. They're very, very small. They're not obstructing anything in terms of fluid flow or anything like that. So you design these things to have the optical characteristic that you want. You design them in such a way that they are of a type of biomolecule that is not recognized as an enemy to the body, so that they don't get engulfed by white blood cells or something. And they just park there. They're just sitting there. So they're dormant until light shines on them. Then they go to work and they do their thing. So anyway, let's see what else he has to say here.
Artificially installed in brain cells so that now we can use that switch to activate or inactivate the brain cell simply by clicking it. And in this case, we click it on with pulses of light.
So we attached this light-sensitive switch of channel rhodopsin to the sensor we've been talking about and inject this into the brain. So whenever a memory is being formed, any active cell for that particular memory will also have this light-sensitive switch installed in, so that we can control these cells by the flipping of laser, just like this one you see.
So let's put all of this to the test.
I wonder if that was a real picture, that thing that was just up on the screen for a second. I wonder if that was, not that one, that's an animation.
Like this one.
That, right there.
Yeah.
Right there.
It's a high-powered laser.
That might be a real image.
But that would be what they put on the head of a mouse, right? So they would install hardware onto the skull of a mouse that would be a mounting fixture for a fiber optic laser to be snapped into place.
That's right. The pointed light source. Exactly.
Mm-hmm. And
okay.
Yeah.
Fascinating.
You see.
So let's put all of this to the test now. What we can do is we can take our mice and then we can put them in a box that looks exactly like this box here. And then we can give them a very mild foot shock so that they form a fear memory of this box. They learn that something bad happened here. Now, with our system, the cells that are active in the hippocampus, in the making of this memory, only those cells will now contain channel rhodopsin.
When you are as small as a mouse, it feels as if the whole world is trying to get you. So your best...
But you know what? Don't be deceived by the scale, because interesting point. A brain neuron in a mouse is about the same size as a brain neuron in a human, except we just have a lot more of them. Apparently we do anyway.
You could also easily research where they take human brain cells, particularly glial cells,
and do it in vitro.
No, they put them into mice, and then the human brain cells overtake the natural mouse brain cells, and so then you have a mouse body with human brain cells inside that they can then experiment on as a mouse.
Yep. And you know what? Short term, the mouse's vascular system will keep those brain cells alive for a good while. Rejection doesn't happen immediately. There's this thing where you take cells from one creature and put them in another one. The immune system gets wind of it and says, you're not part of me. But that doesn't happen right away. They'd have a period of days, at least many hours, and probably days before there would be a defense response from the mouse's immune system to wipe those cells out. And give them plenty of time to study them. Yes. Yep. Where would they get the human brain cells, though? I'm curious.
I don't know, fetal tissue from abortions.
Oh, good.
I got 3 documents that I just clicked on through a search: human glial cells in mouse brain.
Let's come back to. I'll pull this share down. Yeah. Do you want to put the thing up?
Yeah, right now.
All right.
Right now. Here we go. Just so you don't think that I'm just running my mouth, right? So half the cells in this mouse's brain are human.
Holy
Researchers
moly.
implanted immature human brain cells in mouse pups, which then grew and replaced nearly half of the mice's own cells.
Whoa.
A new study. This is December 2014, so we're in the same time frame as Optogenetics development.
Wow.
A new study published in Journal of Neuroscience explores consequence of creating mice that have many mousy neurons but human glial cells.
Okay. The date was 2014.
Yeah.
Oh my goodness. Let's read a little bit of this. Okay.
Yeah,
new study explores. Yeah. You go ahead. Yep.
Please. You have a better radio voice, man. So do it.
Oh no. No.
I'm feeding your ego, Indy. I'm feeding your ego.
New study. Explores the consequences of creating mice that have mousy neurons but human glial cells. Those are the cells that support nerve cells and strengthen the connections between them. Okay, so Steve Goldman, a researcher at the University of Rochester Medical Center, led a team that took immature glial cells from human fetuses, donated to research, and injected them into the brains of the mice. Since the glial cells were immature, perfect. Now I get it. The glial cells are immature TMC, so they're not fully differentiated yet.
Right.
So as they mature, they're now maturing in the environment of the mouse brain.
Right.
And they continue to grow and divide in the mouse brain, crowding out the mouse cell. Well, you know what? You could do that with perhaps, let's say,
turducken.
Dog. Well, no, you could do it with dog glial cells, and you could inject them into a cat's brain, and you could get a cat with a dog brain. You could do that, and you can fill in all the blanks. You could do it. I mean, if they got human cells into a mouse, they could put mouse cells in. Never mind. Anyway, Goldman goes on. It's still a mouse brain, not a human brain. But Goldman told Coughlin, but all the non-neuronal cells are human. So glial cells can mature into the star-shaped glial cells called astrocytes that help to connect the neurons. But human astrocytes, oh, I didn't know this. Human astrocytes form more tendrils and are larger than mouse astrocytes. So you remember when I said that all cells are, cross creature platforms are all about the same size? Well, I'll take that back a little bit. They're probably similar in size, but human astrocytes are a little bit larger than mouse astrocytes. And these are the connectors. These are the connector cells that help to connect the mouse brain cells together. So that difference gives the mice with these human mouse chimera brains an advantage. So you're making a smart mouse. It's like ramping up the power of computer. But what if you were to do the reverse and accidentally, if the mistake was made in a lab and they accidentally, got mouse glial cells and it wound up in like a human thing, and instead you're ramping, the human's brain down instead of up. Mm. I'm just saying, it could be harmful. The altered mice. You think? the altered mice scored higher on a bunch of mouse tests of memory and cognition, including those mentioned above. And I think that the day for mouse intelligence testing is going to come here in the United States. I think that day is coming. All right. This is not the first so-called humanized mouse. It's just the most striking because it involves brain cells. There are mice out there that are mostly mice, but have human immune systems too. Scientists are interested in creating these kinds of animals because although mice make good study animals and are close enough to humans, they aren't a perfect match. Therefore, what they're able to do, since it's actually good for them in research from that standpoint, that they're not a perfect match, because what they can do is see how well they blend together. So the humanized mouse closes the gap a little bit more and can give us better insight into human diseases like cancer, Alzheimer's, heart disease, and more. But in creating mice that are more like us, we're blurring the line between human research and mouse research. These mice have half-human brains and are still very much mice. I mean, a mouse is going to do what a mouse is going to do. There are plans to create rats with human brain cells, as was said. They're moving up to rats. Then they may go to monkeys and other more expensive subjects. But Goldman told New Scientist that he and his colleagues are thinking of putting human cells into monkeys to bring that putting human cells into monkeys would bring up more tricky ethical issues. So they are thinking that it could set some sort of a precedent for blurring the lines even more between human and monkey. It's a big jump to make. If you're talking about a human and a mouse, that's a big jump, and nobody's going to jump over that cavern of ethics and say, whoa, what are you doing? But if you put human brain cells, human glial cells in a monkey, and that monkey winds up smarter than a human, who knows what could happen, because they're also stronger than people. A monkey can break you in half. They're really strong.
Here's another one. Support cells found in human brain make mice smarter. This was 2013, months before. So this is an article that
Nice.
It confirms what the other article was saying. They were the medical doctors looking at this stuff. We don't need to read all this. We're gonna run out of time. This is the one I found this article a long time ago, and the same thing. It talks about how they put them through intelligence tests, and the ones with human brain cells did way better.
What's the date? The date is December 1st, 2014. Okay, got it. Yep. The human mouse with the half brain human. Yep. I mean the smart mouse with the half human brain.
Yep. So you can put 2 and 2 together, folks. I mean, if you think that they've stopped optogenetic testing on the brain of mice, think again. They know very well, in my opinion, they know very well what it does in the human brain.
Well, I mean, things happen, you know. I mean, sometimes, it's not an exact science in the laboratory. Sometimes there's contamination.
Sure, you have a couple drinks, you meet somebody, next thing you know, you're a parent. Yeah, things happen.
I mean, come on,
an accident, you know.
In China, there was a bat that got loose, and that's put the whole world in a tizzy, right? I mean, I don't know what happened.
I call bullshit on that.
Okay.
You know, just saying. We got a little left of that video, or we got a
little bit left of that. Yep, that's fine. Okay. Right here, and then here. You like the way I talk to myself while I'm clicking stuff?
You're fine.
Okay.
There's nothing wrong with a little verbalizing the inner monologue a little bit. Nothing wrong with that.
All right. Well, yep. And we got there.
Response of defense is trying to be undetected. Whenever a mouse is in fear, it will show this very typical behavior by staying at one corner of the box, trying to not move any part of its body. And this posture is called freezing. So if a mouse remembers that something bad happened in this box, and when we put them back into the same box, it will basically show freezing because it doesn't want to be detected by any potential threats in this box.
So you can think of freezing as you're walking down the street, minding your own business, and then out of nowhere, you almost run into an ex-girlfriend.
So, okay.
Or ex-boyfriend. And now those terrifying 2 seconds where you start thinking, what do I do? Do I say hi? Do I shake their hand? Do I turn around and run away? Do I sit here and pretend like I don't exist? Those kind of fleeting thoughts that physically incapacitate you to temporarily give you that deer in headlights look.
However, if you put the mouse in a completely different new box, like the next one, it will not be afraid of this box because there's no reason that it will be afraid of this new environment. But what if we put the mouse in this new box, but at the same time, we activate the fear memory using laser, just like we did before? Are we going to bring back the fear memory for the first box into this completely new environment?
Hey, we're almost out of time. Wouldn't you like to discuss this a little bit? Like, the idea, is there some kind of a fascination about doing this stuff that I'm missing? These are brilliant guys. These guys, and they're dedicated, they're passionate, they're all excited about it. They're saying, yeah, we can do this. We can figure this out. Let's see what happens. It's one of the natural characteristics of humans to like to tinker around with things. But what else could we be doing that would, I don't know, that would, why do all this, I guess? Aren't there shorter routes to helping humanity with research?
You answered your question within the question.
I forgot my question.
You asked why we're doing this.
Okay, yes.
That is the answer. It is to understand why, to figure out the why. That is the answer. That is our nature. That's human nature.
Well,
it's the same general underlying driver that allows us to explore and take risk and all that other stuff. It's just why.
Okay, ask yourself. Why. Okay, fine. I'm on board with you on that. Okay, I'm happy about asking myself why about a lot of things, but they tend to have a different ultimate goal in mind. There's just some things. I mean, I don't know. There is a person who has an interest in any subject that you could possibly name. That's why we're all different, and everybody has a different area of interest and expertise. So these guys are good at this. They know how to work in a lab. They know how to combine things and do all this, and it's backed by a lot of money, and a lot of other people are interested in this too for whatever reason. I don't know exactly what, but I'm not so interested in doing this stuff between this cross species experimentation. I'm not as interested in it personally, and that's just me. I guess. I don't know.
Yeah. Well, at the end of this video, basically they shine light on the mouse while it's in the new box with no environment or with no environmental threats. And then all of a sudden the mouse freezes and freaks out because those neurons have been stimulated with the blue light. That's the end of the video. Basically how the video ends.
And it is, it's 7 minutes away. It's too far. We're only halfway through the video. So, all right. Well, a seemingly innocuous subject, okay? Optogenetics. But look at how many other areas it pulls in with it. And look at the potential on all fronts. And look at the size in the corner of your screen on our stream here right now. You're actually seeing to the side the neuron and the little green implanted little they're the channel rhodopsins. They're there, and they've been made, they've been built, and they are being deployed in lab anyway. I don't know where else they might be deployed. And that is one part about optogenetics. That's one part. There are others, and we're going to get into them one of these days. We do something different every week. And I think that the more that you follow along with us, the more you may find that this stuff is all connected. TMC, thank you for working with me here.
It's always a good time, Indy.
It's quite an exploration. So if you have a little bit of extra time, don't forget, check out humaninternet.org. Also coming up, and it's going to take a little bit of work to get it set up, but I'd like, if you can, anybody viewing, please shoot an email to cyberhumanfrontiers@gmail.com. I will append the link, I'll append the description of the show so that that's down there, because we're looking at streaming out on some other platforms as well. And we want you to be able to find us, and we want to be able to find you. So that's coming hopefully in the near future. And so don't forget, and I've got TMC's links out there to his website, it's really amazing. And thanks for being with us. TMC, what do you think?
I will start working on that website again here very soon, even if it's a little bit every week. I've just had a lot going on, but there's a lot more that need to be added. Like all of this, the whole topic tonight, optogenetics. There's nothing on the site that shares that, where you're able to review it at your own discretion or at your leisure. So I'll make an effort to remedy that. There's quite a few topics that need to be added to that site. So
it's a process.
Yeah. If you have gone there, don't be discouraged if there's nothing new. The information that goes up there is incredibly technical for somebody who doesn't or didn't spend their entire young adult life going to school to learn the nuances of biomolecular technology and neuroscience and everything else. This has all been a self-taught endeavor. So I encourage you all to do the same. Learn something. Pick something of interest and learn it. It's really, really good for your brain.
Yep. Exercise the brain. Absolutely. Well, all right, guys. thank you very much for being with us. And don't forget, if you can shoot an email, I'll make sure that link is up there. It's cyberhumanfrontiers@gmail.com. That way we can get you so that you can find us. I expect that we're probably going to have more extended videos on some other platforms, and that's coming in the future. So thanks everybody, and good night.